Today, we're going to dive into some really cool research that uncovers a kind of hidden gene editor. And it's working inside your cells right now.
So, let's start with a puzzle. Have you ever stopped to wonder why your brain, with all its incredible complexity, is so different from, say, your bicep?
I mean, here's the really wild part. They share the exact same genetic blueprint. A neuron in your brain and a muscle cell in your arm have the same instruction manual, roughly 20,000 genes. So if the instructions are identical, what's the secret? How do you get such completely different outcomes?
Well, it turns out the secret isn't just in the genes themselves, but in how those genes are read and interpreted. Think of it like our body has its own creative editor.
The official name for this process is alternative splicing, and honestly, the best way to get your head around it is to think about a movie editor. A gene is like all the raw footage for a film. Now, the editor can take that same footage and cut it together to make a heart-pounding action movie, or a comedy, or a tearjerker. It's the same raw material, just edited differently. That's exactly what alternative splicing does for a single gene. It creates a whole bunch of different proteins.
And get this isn't some rare, obscure biological trick. It's the main event. It's estimated that a staggering 95% of our multipart genes use alternative splicing. It's absolutely fundamental to how we're built.
Okay, so to really understand how this powerful editor works, we have to zoom in. I mean, way in. We're going to look at one of the simplest genetic switches in one of the simplest animals.
And for that, scientists turned to a tiny, see-through worm called C. elegans. Its simplicity is actually its superpower. By studying these little guys, we can uncover these really basic rules of biology that, believe it or not, often apply all the way up the evolutionary ladder to us.
So researchers zeroed in on one particular gene in this worm, a gene called ZOO1. And they noticed something fascinating. This one gene was being read totally differently depending on what kind of cell it was in. Here's exactly what they saw. There's a specific chunk of this gene, called exon 9, that acts just like a light switch. In the worm's neurons, that chunk of genetic code gets snipped out. It's skipped entirely. But in the muscle cells, that exact same piece is kept in. It's included. It's a perfect on-off binary switch.
Which of course leads to the million-dollar question that kicked off this whole investigation. What or who is flipping this genetic switch?
And that brings us to our prime suspect, a molecule that seems to be the master regulator pulling all the strings behind the scenes. Let's meet our culprit. It's a protein called UNC75. In this little scientific detective story, this is the guy that was seen at the scene of the crime, and the evidence against it is starting to look pretty convincing. So, let's look at the case against UNC75. First off, it has the right job description. It's an RNA-binding protein, which is exactly the kind of molecule you'd expect to be controlling splicing. Second, its location is suspicious. It's found in neurons, the very cells where the exon is skipped. And third, the alibi is perfect. It's completely missing from muscle cells, where the exon is included. I mean, it's the perfect candidate.
So we have a suspect, a motive, and opportunity. But this is science, right? You need more than a hunch. You need cold, hard proof. It's time to run the experiments and crack this code.
Okay, here's the first big experiment. Scientists went into the neurons and just got rid of UNC75. And look what happened. The switch completely flips. The exon goes from being skipped almost all the time to being included almost all the time. This is huge. It proves that UNC75 is necessary to make the neuron skip that exon.
But here's the real smoking gun. What if you do the reverse? What happens if you take a muscle cell, which normally doesn't have UNC75, and you force it to start making it? The result is just, wow. The exon inclusion rate plummets from over 90% down to 0. The muscle cell starts editing genes just like a neuron. This proves UNC75 isn't just necessary, it's sufficient. It can flip the switch all by itself.
So how does it actually do it? The mechanism is surprisingly simple and physical. First, UNC75 latches onto the RNA message at these specific docking sites, right next to the exon. Then, just by being there, it acts like a roadblock. It physically blocks the cell's natural splicing machinery from getting in, and if the machinery can't see the exon, it just skips right over it. It's simple, it's elegant, and it's incredibly effective.
Okay, so a tiny worm, a single protein, a genetic switch. It's all fascinating stuff, but what does it all mean? Let's zoom back out and see why this tiny discovery has such big implications for us.
The researchers themselves summed it up perfectly in their paper. What they essentially said was, hey, we've shown that by arranging these protein docking sites in a certain way, you can create a super reliable on-off switch for a gene, and you can make it work inside a living animal.
So why should you care about a protein in a worm? Well, here are the big takeaways. First, this reveals a fundamental rule of biology. These master regulator proteins, like UNC75, are a key part of what makes a brain cell or a muscle cell a muscle cell. Second, this isn't just a worm thing. UNC75 is part of a whole family of proteins called CLF proteins, a family that we have in our bodies too. And that leads to the third and most important point. This has direct links to human health. When our own CLF proteins don't work right, they're linked to some serious neurological and muscular disorders.
And all of this brings us to one final, powerful question. This research, which started by looking at a tiny worm, has uncovered a fundamental switch that controls what our cells become. So, is it possible that truly understanding these master regulators, how they work, why they break, and how to fix them, could be the key to developing new therapies for some of our most complex and difficult diseases? The research is still unfolding, but for the first time, it feels like we're finally on the right track.